You're listening to the David Feldman radio program. You sad, pathetic hump.
The irritable immunologist is back. He's talking to us from a lab somewhere inside the La Jolla Medical Center Research Complex. Hello, Irritable.
Hey, Dave. How you doing? Still living?
I'm still living. I'm in New York City, and it's pretty terrifying. You were right. You were right about this.
I think a lot of people were right. Unfortunately. But,
yeah.
So you guys are edging up near 1,000 deaths directly as a result of the COVID-19 outbreak. Yes. Yes,
and there are reports already that the emergency rooms are overflowing, and Bill de Blasio says he has about a week's worth of medical supplies left for the city of New York.
Oh, God.
Yeah.
Oh, my.
Yeah.
Well, yes, this is going to be a very bad situation. Hopefully some of the sheltering in place orders will reduce the number of people who are not seen by medical professionals. I think it's probably real important to note. I've seen a lot of commentary, particularly from econ types and hedge fund manager types, about how this is just X additional number of dead people, and I don't think those people really quite grasp what having an overflowing emergency medical system means. It means they don't have any surge capacity. It means that if you break your knee and you're hemorrhaging, you're going to have a problem getting into the ER because it's completely packed full of people who have a highly infectious, potentially fatal coronavirus infection. So, yeah, I think...
Because we've had senators, Senator Ron Johnson, the Republican, wrote a piece in USA Today saying we're overreacting, that the cure can be worse than the disease, and we have to learn to live with a certain amount of risk and death. And he said something like 50,000 Americans die from suicide, 70,000 die from drug overdoses. Get over it.
Sure.
But I'm looking at the emergency rooms. I don't see the emergency rooms overflowing with suicide victims and drug overdoses.
Indeed so, and I mean, perhaps what Senator Johnson is failing to understand is that this is above and beyond the pre-existing steady state death rates of Americans from all causes who have to go to the hospital. I find this particularly tragic for people who have to go through chemotherapy, and especially the nurses who do the infusions at the infusion centers. Hopefully, hospital managers are trying to keep those people strictly segregated, because anybody who has a pre-existing medical condition which messes with their immune system is going to be at significant risk here. And the nurses who treat them are going to be at significant risk of passing it on to people who can't fight it off effectively. So, yeah, I think Mr. Johnson and many other people are really not quite grasping all that this implies, to just say, hey, it's okay, let's all go outside and play on the beach and let the chips fall where they may. And by chips, I mean elderly people or children with cancer.
Right.
I don't think that's a good call. And I think there's certain specific attributes to coronaviruses in particular that make that attitude especially unfortunate. This is not influenza. This is not something that has ever had an effective vaccine developed for human beings for any coronavirus. Right. There's at least 4, as I've talked about with you before, that are in constant circulation. Vaccine never developed. There were trial vaccine attempts in animal models for the original SARS virus, which is the most closely related virus that infects humans to this one, known at least. And those trials ended in disaster, insofar as it appears as if SARS, and probably this virus as well, can engage in what's called ADE, or antibody-dependent entry. And what that means is if you don't get your vaccine exactly right, if you have what's called cross-reaction with a closely related species, the presence of low levels of antibodies to this virus will actually help it infect the body. In order to prevent it from infecting, you need high titers, is what they would call that in the blood.
High what?
Antibody specific.
High what?
Titer. 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 Not any sort of sick or twisted sexual fantasy, Dave. I realize you prefer to go off on that tangent, but not today.
Yeah.
So yeah, there's
so let me see if I understand what you're saying. First, you're saying that let's say 2 million Americans die every year. We will get a number when this is over, and they'll say 100,000 people died from the coronavirus. What they won't tell us is that a lot of people died not from the coronavirus, but we had a public health system that was so stressed out it couldn't accommodate children with cancer, heart attacks, that the surge of patients ended up killing people. But they will not be listed as dead from coronavirus. They'll say heart attack, cancer, untreated because of the coronavirus. That's right. One thing you've said, and that's what they're not talking about right now. Good luck going to an emergency room in Manhattan. Good luck if you twist your ankle. Secondly, what you're saying is that they did try to come up with a vaccine for SARS and MERS, and they weren't able to do it?
The trial vaccine that I'm aware of, I'm not sure about the case of MERS. I think that was not heavily investigated because it was a much more limited number of people who contracted it. But yes, with SARS in the animal model, which I believe this was in both mice and ferrets. I'd have to double check. But they gave them this, it's a protein vaccine trying to induce the immune system to react to the virus's protein by just injecting, parts of those proteins or just the proteins themselves, not the whole virus. And they found in those animal trials something called antibody-dependent entry. So the presence of the vaccine in some of the trials for SARS failed, and failed to the extent that it was possibly worse than no vaccine at all. Now, this doesn't mean that a vaccine is impossible. The Moderna, I believe, and other groups are trialing what's called mRNA vaccines, which work a little bit differently. You're inducing the body to produce this protein itself. You're not just injecting it with something called an adjuvant. So the methodology is quite different, and this may work out. There appears to be a lot of faith in the ability to design effective vaccines against coronavirus, and that just hasn't been done in humans. And it's been tried extensively in cats as well. There's a coronavirus that infects a large percentage of house cats, and sometimes it turns into something called FIP, which is almost invariably fatal. The coronavirus gets out of hand, starts infecting cells of the immune system, and kills the cat. There's been vaccine trials for that. Same result as with the SARS vaccine trials in ferrets, which was the vaccine itself. If it didn't produce enough specific antibody, and when I say specific, I mean very specific, to this virus, it actually helped the virus. If it had mutated even a little bit, or the fit between the antibody and its target, called the epitope, wasn't exactly perfect, it made the infection worse in cats. So there still is, to my understanding, no vaccine for FIP in cats, which is, again, caused by a coronavirus. Coronaviruses are really tricky. So there's mRNA vaccines. There's some really serious advances in terms of, we'll call it glycoprotein immunology. That is proteins that have sugars attached to them. There are groups that specialize in just this, and that may be a very good target for coronaviruses. But as to this point, assuming that we can get a quality, responsive, good long-term antibody titer that's going to counteract this particular coronavirus right now, nobody's been able to demonstrate that in any coronavirus in humans.
Okay, so let me just circle back and try to unpack what you've just told me. SARS and MERS are coronaviruses, correct?
Yes.
Okay, and they are respiratory illnesses?
Yeah, they're both respiratory infections, huh?
Are all coronaviruses respiratory?
No, a lot of them are gastrointestinal. In pigs where there's a trial vaccine that appears to work for a gastrointestinal coronavirus, that's for PEDV. To my knowledge, that's the only experimental vaccine that I've seen efficacy in an animal model. And that was, I believe that was even doing a super roundabout way where you were vaccinating the mother, the sow, and then the piglet would consume a particular type of antibody that's secreted in the mother's milk called secretory IgA. And that was shown to be effective.
Now, SARS.
To my knowledge,
okay.
Go ahead.
So SARS came to us from Asia. MERS came to us from the Middle East.
Yeah.
MERS comes to us from camels. SARS comes to us from pigs. Or
bats.
Bats.
SARS is almost certainly bats as well. Yeah.
Okay, bats. And SARS and MERS were pandemics or epidemics?
I'm not. The WHO has semi-recently changed its designation on this. I don't believe they were considered true pandemics. Possibly SARS-1 was. MERS, I don't think there were enough patients to consider it that, but it was definitely multinational.
Okay. So had SARS and MERS become pandemics, would we have been able to come up with a vaccine?
It's a good question. I mean, that's why I'm mentioning this in case people are reading sort of even popular science reporting or, more especially popular media reporting where they're like, oh, we'll have a vaccine out in 18 months. That could be. The problem is just that just hasn't been done for any human respiratory coronaviruses, and 229E has been known about for probably 50 years. OC43, probably nearly that long. They've been in the human population for centuries, coming over from possibly cattle or horses. And
who dies? I mean, are you more likely to die from the influenza or from SARS and MERS? I mean, we seem to have a vaccine for the influenza every year. I don't know how effective it is, but
Right, that's variable, for sure. They do a really amazing job formulating the vaccine, and they do that based on surveillance, particularly in Asia, of what the strains are in the animals that influenza can infect them as well as humans. But yeah, they're not perfect, but that's a really long established model and can work pretty well. SARS and MERS, SARS-1 had at least a case fatality rate of about 10%. So it was quite a bit more deadly than this. I think MERS was close to 30, if not more.
30% one case morbidity.
Case fatality, yeah, exactly. So people have the virus and they died of it.
In the hospital.
They were usually, yeah. I mean, they would have to have it detected.
But other people could have SARS or MERS, and it's a mild case.
I mean, possibly. I'm definitely not an expert on what the clinical presentation of a mild case of MERS would be.
So why didn't they come up? Why didn't they come up with a vaccine? Was it just dying off by itself? What ultimately stopped?
Containment worked in both those cases. Containment worked once they figured out what was happening. MERS is probably much more difficult to transmit person to person. So I think a lot of the cases in Korea in particular were from like one guy who was, I believe, a businessman from possibly Saudi Arabia or Kuwait, who came in and sort of spread it about because he kept going to hospitals and they couldn't figure out what was wrong with him. So he spread it to multiple hospitals. But other than that, MERS is typically not person to person. SARS certainly was a bit, but in both cases, the national and the international health community freaked out enough and got enough on the ball early enough to contain them. So they're not stretched across the entirety of the human population like the 4 or 5 endemic common cold type coronaviruses. I think with this virus, so many people have it in so many different countries that there's a real possibility containment may be nearly impossible at this point.
And so containment is nearly impossible, so it gets down to treatment.
Yeah.
Treatment. Now, there was a story I saw in the Wall Street Journal. We're being told not to experiment with anti-malarial drugs. There are people.
Sure, don't do that. Don't do that by yourself.
Don't do it.
Yeah.
Never
do that.
Hydroxychloroquine. Is that the anti-malarial drug that somebody took as a preventative and ended up killing themselves?
They may have gone with straight chloroquine. Hydroxychloroquine's a little less. The therapeutic index is more broad. The persistence in tissue is even longer, though. So I believe that was chloroquine phosphate from like aquarium for using against fish parasites, and they took way too much and killed themselves. Yeah.
Okay, so Donald Trump, again, he's the worst possible man to be our president right now. He has spoken optimistically of hydroxychloroquine, an anti-malarial drug. There are people who say that a combination of hydroxychloroquine and azithromycin together can, as both a preventative measure and a treatment, can work. What are your thoughts on that?
I mean, the clinical data, there's fairly decent clinical data to suggest for people who have a serious case that combo might be helpful. The main study that's been published that is very ad hoc, let's put it that way. So the level of control that they had in that study was not super great. And in their principal experimental group where they combined hydroxychloroquine and azithromycin, I believe it started out with an N value. The number of people enrolled was 16, and it ended up with 6 because 10 of them dropped out. So that's not good study design.
Right.
So having real solid conclusions about the value of these antimalarials seems still a little bit early. But yeah, they're definitely promising. Again, most antimalarials are quite toxic. Z-Pak, azithromycin, isn't an antimalarial. It's an antibacterial.
Malaria is a parasite?
Yes. So malaria is much more closely related to you in terms of things that can infect you than a bacteria or a virus. So the more closely related the thing that's causing an infection in you is to you, the harder it is to treat that, because you need to find something that will impact something related to you and not you, which is a much easier thing to do with bacteria, especially, and viruses.
Let me just circle back. So a parasite is a living, breathing thing similar to you. So whatever medication...
So the malaria is a protozoal parasite. So it basically likes to... A mosquito will... Okay, so let's start it this way. There is, in mosquitoes all across the world, David, and I know this will come as a shock to you, an enormous amount of parasite sex happening in the guts of those mosquitoes.
Sex.
And once they've done their dirty deeds inside the mosquito gut, and the mosquito bites you, it will inject a specific form of this parasite into your bloodstream. That will go in, colonize your liver, and then start, let's just say, go through a maturation and reproduction process. And some of the individual protozoa will get out and basically go into your red blood cells and start chewing them up on the inside. And so chloroquine and derivatives, quinines, form quinolones. There's another one that the Russians are claiming is very functional that causes serious psychiatric side effects. All of these are attempting to target this parasite that is a eukaryote. So it's much more closely related to animals and plants and fungi, for that matter, than a bacteria.
Yeah, define eukaryote. You mentioned eukaryotes before. What is a eukaryote?
That basically, it means a cell that has a nucleus is sort of the most fundamental functional differentiation. And a nucleus is a separate bag inside of your cells that contains the overwhelming majority of your DNA. This is where your chromosomes are kept. And they're sort of segregated from the rest rest of the inside of your cell. And so eukaryotes are all cells or things comprised of cells that have a nucleus inside.
So let me guess. Am I going to guess that the coronavirus is not a eukaryote?
No, it's a virus. In fact, it's incredibly distant in terms of its relation to you because it's an RNA virus.
Right.
So its genome is single-stranded RNA, which is quite crazily different.
And so the treatment for the coronavirus, as I understand what you're saying, should be easier since you're killing something that isn't close to you. That
Yes and no. Yes, in terms of I've talked to you a bit about it before, and remdesivir, which is an experimental anti-Ebola drug that didn't work that great against Ebola, but probably works fairly well against this, probably. That's an antiviral drug, yeah, and that targets the virus pretty well. It's basically poisoning an enzyme that the virus needs to replicate itself. It's RNA-directed RNA polymerase. You'll see it mentioned now and then as RDR-POL. So remdesivir, and there's another one, Fujifilm, has an anti-influenza drug that's trying to do something similar that's poisoning enzymes that viruses use, that you don't have an exact copy of that. And so if it was poisoning an enzyme that malaria used, very likely malaria would be using Plasmodium, the genus of the protist that causes malaria, would be using enzymes that are much more similar to yours. And so, yes, the antiviral drug design is trying to target specifically viral things without hurting you very much, which I think has been a great success, particularly for what's called HAART therapy for HIV, highly active antiretroviral therapy. But the downside with antivirals and viruses is viruses mutate extremely rapidly. And this is a group 4 virus. This is a single-stranded positive sense RNA virus, and that group of viruses tends to mutate faster than any other group of viruses, unfortunately.
You're saying that the coronavirus mutates?
Yes, all viruses mutate.
But this one
can't be got around, but
this one mutates more than others.
Mutates more than a lot. Yes,
Now, last time you said it could mutate to a safer type of virus.
Yeah, that's theoretically possible. If it's in circulation for long enough, the fatality rate may drop over time if it becomes better adapted to its host. It could go the other way, too. But that would certainly be long-term evolutionarily favored. Yeah. The thing is if one coronavirus picks up 2 mutations, one which helps it spread better, and the other which kills more people, if it can spread better than its close relatives, that may outweigh its fatality, limiting its spread by killing its hosts more rapidly. So I don't think anyone should be making really serious predictions on this, but yeah, RNA viruses mutate like mad. Single-stranded positive sense RNA viruses mutate faster than any other kind of virus. Coronaviruses mutate more slowly than other viruses in this group, happily.
Okay,
so they actually do have some error correction.
Okay, so in terms of evolutionary imperative, the virus doesn't want to kill the host, does it?
The virus wants to have the highest possible fitness relative to all conspecifics, if a virus could want anything. So it wants to be able to, what's going to be favored is its ability to reproduce differentially more than similar viruses. So it's in competition with its close relatives to infect as many people as possible, as quickly as possible. Now, with viruses, this gets very, very complex because sometimes multiple viruses like to team up. So there's some evidence that respiratory syncytial virus, RSV, likes to team up with coronaviruses now and then. But yeah, in general, this is the way evolution works, is the higher the fitness of a given individual in a population, that means the larger number of offspring they will have in the subsequent generation. And over multi-generational time, the individuals that have the genes that are reproduced the most will have the highest fitness.
Okay.
So, yeah.
So,
You need any more really complex concepts?
Well, we're going to learn this.
10 seconds?
We're going to learn this, Irritable. We're talking with the Irritable immunologist. He's talking to us from a lab somewhere in the La Research Medical Center complex, Southern California. I'm sorry?
I was just making irritable noises, Dave. Go ahead.
Oh, okay. Herd immunity. Any chance that kicks in? What is herd immunity? And
Sure. Yeah. So I think you could certainly ask epidemiologists about this, but essentially herd immunity is when a significant enough percentage of the overall population has been exposed to a pathogen and fought it off, and then was able to generate memory immunity as a consequence of that. The higher the percentage of people who are immune to any given virus or other pathogen, the more difficult time that virus will have transmitting itself. If I'm 2 feet away from someone who's 2 feet away from you, and I have the coronavirus, and the person in between us has already had it and has functional memory immunity, it's quite possible they will be able to short-circuit this. Now, this isn't an instantaneous interaction. Let's say I'm coughing on someone who's 2 feet away from me, and then a couple days later they go over to your house and cough on you. If they've already been exposed, they may already have fought that virus off because the production of antibody and a T-cell mediated response as well as other responses in the body is much more rapid the second time you're hit with a given pathogen. Again, this is a coronavirus, though. So coronaviruses tend to circulate through the human population every 2 or 3 years. And so cross-reactivity in terms of antibodies this is another complex concept. But essentially, coronaviruses mutate fast enough that it may be possible that herd immunity won't count for much in just a handful of years if they mutate rapidly enough to alter what's called their serotype.
Okay, so let me circle back so I understand this. You get you're exposed to the coronavirus. Everybody who's exposed to the coronavirus and survives develops antibodies. If the coronavirus...
T-cell responses,
I'm sorry?
Yes, in T-cell responses, exactly.
And once you have those antibodies, do you have them... I think you told me you don't have them forever.
So, yes, this is one of my concerns with coronavirus vaccine design, which I'm not doing. But yes, long-term immunity to coronaviruses in the few publications that have looked into it suggest that response, the antibody response, tails off over time.
That's not about mutation. This has nothing to do with the mutation. This is the same virus.
Yeah.
If it makes a comeback, even though you have the antibodies, those antibodies become weaker and or disappear.
They become less of them over time.
Okay.
In terms of the titer of antibody in your blood. But again, the coronaviruses are almost always going to be something of a moving target because they can mutate pretty quickly.
And once they mutate, your antibodies that you have
may not help you. Yeah.
May not or won't help you.
May not. It depends on where the virus is mutated. So the antibodies are going to react principally with proteins exposed on the surface of the virus. So like the spike protein, which sits out a bit, and or bits of the actual virus itself cut up and presented to cells in a certain way for a T-cell response. So coronaviruses can mutate pretty quickly, and so given that a fair number of them, 4 or 5, are implicated in causing, some people will say 15, some people will say up to 30% of common colds, they seem to come back around every few years. Some of these strains that have been in the human population for a long time. So herd immunity, at least natural immunity, may not be that great for this virus. We just don't know.
Right.
And so, you might want to ask epidemiologists in particular for their thoughts on this. But, it's...
Without going into too much detail before you go, you are working on a vaccine, you personally. Is that correct?
No, we're on an antiviral drug. antiviral drug design, not a vaccine. No.
But you are. Oh, so you're working on a treatment for the coronavirus.
We have potential. There's lots of people in any major biomedical research complex in any of the main areas in the United States and worldwide who will be working on pretty much exactly what we're doing, which is desperately trying to find either previously FDA-approved drugs or novel compounds that are from super bizarre sources that have never been tested. And so, I'm certainly involved in screening things to see if they're functional against this particular coronavirus.
Last question, and I hope you'll come back for Friday's show. I have a lot of reading to do about biology. I have to remember my biology, and I have a lot of reading to do. We get SARS, you say, from bats, MERS from camels. We're now...
Yeah, I think dromedaries, but yeah, that's camel. Go ahead.
And we're getting reports that cats carry? I hate to even go here. Is it possible that cats are carrying the coronavirus? They do die from
Probably not this one. So that's a different virus. That's not this. That's not SARS-CoV-2. That's not COVID-19. That's a different coronavirus that causes something called FIP when things go wrong for whatever reason. Yeah. So there's different coronaviruses that infect all kinds of animals. Bats in particular, viruses love to target for very interesting reasons about bat biology. They don't really have bone marrow, so their B cells aren't exactly B cells, so their antibody response isn't the same as most other mammals because they have more or less hollow bones in order to fly, and that's where your B cells mature. And fundamentally, that's where all of your immune system comes from, is from the hematopoietic system in the bone marrow.
Okay.
So, yeah, I wouldn't... There are molecular testing services for pets, and one of them, I think in Maine, perhaps, one of the big ones, has screened a ton of animal pet samples for SARS-CoV-2, and I believe they've been entirely negative, which makes sense. I wouldn't expect any normal pet to contract this. There is one exception, however. The probable animal model that will be used for a lot of research will be the Syrian hamster. That appears to be, it shares a lot of similarity in terms of its ACE2 structurally. So it's possible if you have a Syrian hamster, they might be able to contract this. But if you've had them for more than a couple of months, most likely they're going to contract it from you rather than the other way around. So, yeah, I wouldn't worry too much about your pets. There are other coronaviruses that infect most types of mammals, but not this one.
Okay. I've got a lot of reading to do. The irritable immunologist...
Oh, really quick, David. If you have anybody who's a retired health professional or health-associated professional, there's a federal health professionals registration, and that includes veterinarians, EMTs, and dentists. Anybody who has good clinical training of any kind, it's called the ESAR-VHP, the Health Professionals Registration. Also, if you're in harder-hit areas, like in New York City or out here in California, if you have a supply of disposable, preferably nitrile gloves that are unopened, consider donating those to the local whoever, local medical clinic, the local janitorial service, anybody who's essential. They're going to need personal protective equipment. So if you've got masks, if you've got gloves, and you think you can get by without using them, look into donating locally.
What about my S&M mask?
I don't know if that's N95 certified. Did you get a proper fitting? Was your dominatrix wearing a lab coat? I guess that's the real question. But yeah. Also, definitely call in if you're at home and you're just antsy and you want something to do, something productive. Go ahead and call the House of Representatives and Senate principal office, which is 202-224-3121.
Okay.
And or go to house.gov or senate.gov and try to contact your representative and say, hey, we need to cover all medical expenses during this pandemic.
Yes.
Not just for this, all medical expenses, because anybody who needs medical treatment is going to be impacted. Also, as I mentioned last time, if people could underline the importance of getting homeless people off the streets and or getting hand washing stations and bathrooms put out in the streets so that people have some way to clean, especially their hands, that's going to pay dividends more than it costs. Because a lot of the people who are out on the street are, relying on stores that are closed right now to go in and wash their hands when they can. And so I'd really like to emphasize that for people as well. And, the more pressure you can put on your representative, be polite, but the more pressure you can put on them to say, hey, we can do more massive testing than we're currently doing. We have other resources, academic and research resources that could be used to do much more massive testing, at least for the state epidemiology people to look at, to give them some kind of data, because right now we just don't know how many people are infected in this country, and we're not ramping up testing fast enough. You'll see a news story every day about X company or Y company. There's at least 4 down here who have special custom machines. They will sell you a super quick SARS-CoV-2 COVID-19 test, and that works really great unless you don't have that machine. And they probably don't have 5 million of those machines stockpiled. So, again, there's lots of resources that could be leveraged if HIPAA requirements were slightly relaxed, if some of the health privacy stuff was done at a central location, and then you feed anonymized samples out to labs like the one I'm in, and a lot of the ones that are idling right now. They can do RT-PCR. And if you feed them RNA or cDNA, which is what you make from RNA before you throw it into an RT-PCR machine, they can handle that safely. That's not infection.
Right.
So, again, definitely call your representative, say, hey, we need 6 months guaranteed unemployment insurance for everyone. Having 3 million people go unemployed in a matter of a couple of weeks is madness. And we need to have all medical expenses covered for all Americans for at least a 6-month period.
Well said. Well said. You'll come back Friday.
Yeah, man. I mean, I'm working 7-day weeks here, but I'll see what I can do. We'll make some time.
The irritable immunologist joined us today, and he'll be back later in the show as Jackie the Joke Man Martling.
So this Chinese guy and a rabbi
All right,
and a priest walk into a bar with a horse.
Okay, buddy. I'll talk to you on Friday. Thank you.
All right. Take care, everybody. Be safe.
Thank you.